everyone. We left off last time in the first protein synthesis PowerPoint um, discussing how proteins are made using the instructions in DNA, and that DNA, the sequence of the bases, the ATGC in the DNA, gives the instructions for which amino acid is to be put into a protein when it's being synthesized on the ribosome. And so here is your DNA. It's double-stranded. One side is used for protein synthesis. It's called the template strand. And in the nucleus of the cell, that side is used to make complementary mRNA, messenger RNA. There should be an A next to that. And so if there's a T in the DNA, there's an A in the RNA, a C, a G, so forth. And so at the end of transcription, you've made an RNA that's complementary to your gene, your DNA. So if I told you the sequence of this RNA, you would be able to tell me the sequence of the DNA because of the matching bases. They don't have it in here, unfortunately, but if there were an A in the DNA, it would be a U in the RNA. And then every three of these bases in the RNA represents a particular amino acid. And so the ribosome knows which amino acid goes in a particular spot because of the three codons that represent, the codon that represents the amino acid for that spot in the protein. The message of which codon represents which amino acid is something called the genetic code. We mentioned this last time a little bit, and the genetic code is this table shows you what the genetic code is. You don't have little tables inside of your cell. The way it actually happens inside of your cell is by what molecules fit with what molecules and can bind together. But suppose you have a codon. G, C, A are the three bases in your codon on your messenger RNA. So this gray thing here represents the messenger RNA that was made from the DNA. So the codon is GCA, and this table here shows you the kind of translation process. And the way it works, the first base, you go down here. So our first base is G, so that means we're going to be in this section of the table. Our second base is C, so that means we're going to be in this section of the table. And then our third base is A, which is this one right here. So GCA in your messenger RNA represents the amino acid alanine. So whenever on the ribosome you're making a protein and the messenger RNA has a GCA, the amino acid alanine is put into the protein. And I mentioned that's a universal code that occurs for all organisms on Earth, that whenever there's a GCA in the RNA, the amino acid alanine. This shows you all the different codons and which amino acid. You can kind of see from here that some amino acids have more than one codon. This is leucine here. Leucine actually has 1, 2, 3, 4, 5, 6 codons. Phenylalanine has 2. So it varies a little bit. The first codon is always AUG. That's called the start codon. And then there are 3 stop codons, UAA, UAG, and UGA. So when that gets encountered by the ribosome making the protein, either of these 3, that means that's the end of that protein and protein synthesis stops. So the AGU shows where the beginning is. Then there would be all the codons for amino acids. If there were 31 amino acids in the protein, there would be 31 codons. And then at the end would be a stop codon as a signal that that was the end. So when you have your RNA, you would have a string of letters or bases, and every 3 of those represents a codon, which then represents an amino acid. And that's how the DNA gets changed to RNA, which then is used to give the instructions for what amino acids you need to make your protein. This is just a little practice one. So you would need a copy of that genetic code that's on the other slide to do this. So it'd be nice if you pause the PowerPoint right here. And these are your 4 bases in DNA. So the first thing you have to do is make your RNA. And then you would look up each RNA codon on the table that was on the slide previous. When I'm narrating, I'm not able to go backwards, and look that up to see what the amino acid is represented by that codon. For example, TAC would be A, and A goes with T if it's DNA, U if it's, and C goes with G. So the first C codon in this protein, this is only 4 codons, so it's probably not really a protein, but I just made these letters up, is AUG, which is the start codon. So you would do all 4 of these to get what the mRNA is and then what the amino acids are. So this is the answer to that. If you tried it, you can check and see if you got it right. So TAC in the DNA becomes AUG in the RNA. And if you look that up on the table, that's methionine, which is the start codon. And then GCA becomes CGU, because G goes to C, C to G, A to U. If you look that up in the table, it's arginine, and so forth. TGG would be ACC, that's threonine. AAT would be UUA, and that's leucine. So that tells you how you can figure out what amino acids are made up by a protein using the codes in first DNA and then RNA. So again, transcription is the first part of making a protein, and that's the part of making the messenger RNA. And it's called messenger RNA because it has the message of what codons. So you have your DNA double helix. You unzip the part for the gene, like if you need more insulin, you would unzip the insulin gene. And through transcription, you would make RNA. So this blue line here is the RNA. So this is the DNA. And again, the RNA would be the complementary bases. So G goes to C, CG, CG, etc. And then each of these 3 letters are a codon, which would represent an amino acid. So this process is called transcription, of making the RNA using the base sequence in the DNA. That happens in the nucleus. And at the end of this transcription part, there's some processing done to the RNA before it actually leaves the nucleus. We're not going to really go into the details of that, but at the end of transcription, the RNA will leave the nucleus and go out to the ribosome. And this is just showing a blow up. So here would be your gene that is unzipped so it's not twisted up. And then you're making messenger RNA, which is the green part here, using the bases in your DNA. The enzyme that does that is called RNA polymerase, which kind of makes sense. It's it's making the RNA polymer. And so that occurs inside of your nucleus. Okay, the second step is translation. And translation is when you take that messenger RNA and use it to make your protein. So you have this piece of RNA that comes from the nucleus that has the start codon, then all the codons for the amino acids, and then it has a stop codon. And that is going to give the instructions on how to make the particular protein that you're making. To do this, we need a couple other types of RNA, which I'm going to explain as we go through. Let me go to the next slide. There's actually 3 different kinds of RNA. The one that is made in transcription is called messenger RNA, and it has all the codons for making your protein that you would look up on the genetic code to see what the amino acid is. So when you have a codon, so say we look at this codon here, CUU, and if we look that up on this table, we would see that CUU requires the amino acid leucine to be added to the protein. So you need a way of bringing that amino acids. You eat proteins, so inside of all of your cells are all the proteins, all the amino acids from the proteins that you've eaten, and they're there to be used for protein synthesis. And so floating around inside of your cytoplasm is the amino acid leucine. And when you need that amino acid to make a protein, it attaches to another kind of RNA, which is called tRNA. And it's this kind of squiggly shaped thing here. And if you think the T stands for transfer, and what it transfers is the amino acid. So here is the messenger RNA. It says, oh, we need a leucine now to make our protein. The tRNA has a leucine attached at one end, and the other end has what's called an anticodon, which matches the codon. So our codon, we said, was CUU. So the anticodon is GAA, and they will hook together right here. Oops, I'm sorry, didn't mean to erase that. And at the other end is the amino acid, and that can be attached to all the other amino acids used in making that protein. So that's the second type of RNA that's used in protein synthesis. So first we had the mRNA, which is made in the nucleus through transcription. Then we have the tRNA that brings the amino acid. And the third type of RNA is actually the ribosome. So remember how we said the ribosome is where in the cytoplasm, that's where your proteins are made. It's this little structure inside. And that is made up of the third type of RNA, which is called rRNA. And the R stands for ribosomal RNA. So we have, coming from the nucleus, the messenger RNA. It attaches to the ribosome, which is this kind of peachy colored thing. And then the tRNAs, which are these here, are bringing the amino acids. So the first codon is AUG, which is the start codon. So this, and it also codes for the amino acid methionine, which is always the first amino acid. So this is the tRNA, has an anticodon for AUG, which is UAC. On the other end, it has methionine. So it brings methionine. Then the next codon is UUC on the messenger RNA. It codes for the amino acid phenylalanine. So the transfer RNA brings phenylalanine. It attaches with its anticodon. So U goes to A, U to A, C to G. And now the methionine and phenylalanine are next to each other, so they can form a peptide bond. And you have your first 2 amino acids in your protein. Then the mRNA kind of shifts down. That first transfer RNA you don't need anymore because the amino acid now is attached to phenylalanine, so it leaves. And then you go to the next codon, and for this one, it's CGA. If you look that up in the genetic code table, it stands for the amino acid arginine. And usually when you're writing the amino acids, you just use the first 3 letters. And so this is the tRNA for arginine. And it has the matching bases, C to G, G to C, A to U. It brings arginine. That's going to now hook on to phenylalanine. Then that'll shift down again. And you keep doing that until you reach a stop codon. Then when you reach a stop codon, the whole thing kind of falls apart and your protein is done and the process is stopped. This is just another picture. This is the tRNA bringing the amino acid. And so you keep making your protein, and however many amino acids is in your protein, that would be how many you keep adding before you reach the start codon. Insulin has about 31 amino acids. So you would keep going, and after you got the 31 amino acids there, you would get the stop codon and you would stop. Hemoglobin has about 130 amino acids, so it's a much bigger protein. So it would take longer to make that, and you would have many more codons, but eventually you would reach a stop codon there too. This is just a summary that transcription is in the nucleus. Oops, let me switch to. And it takes the DNA and makes mRNA. This is the processing. You don't need to worry about that. Then the mRNA leaves the nucleus through one of those little nuclear pores in the membrane, comes out, attaches to the ribosome, and then the protein is made. Now, we've talked throughout the course about mutations. We talked about genetic mutations, things like sickle cell anemia and PKU and cystic fibrosis. And a genetic mutation, technically what a mutation in a gene is something that changes the DNA of that gene. So most commonly, it's a change in the base sequence. And that can result in an abnormality where the protein is not made properly. It doesn't have to. So with a mutation, you get a change in your DNA. That can be a good thing. It can make whatever that protein does work even better. Or it can be harmful. We tend to only talk about the harmful ones, but I would say if we were in class, you could look around the room, and the reason we don't all look the same is that we have different mutations, which results in different gene expression, and that's why we all look differently. So we're going to talk about some types of mutations. I'm going to talk about these that are called point mutations, which means it's just a change in one base. So say if it was supposed to be ATC and it becomes AGC, that would be a point mutation because it's just one point in the DNA that is changed. So we're going to talk about different types of point mutations. So in this slide, this one is considered the normal here. Okay, so this would be the normal gene. So this is DNA, RNA, and then the amino acid. So in the normal gene that has not mutated, one of the codons should be ATG. And when you transcribe that to make messenger RNA, A becomes U, T becomes A, G becomes C. So the messenger RNA, the codon is UAC. And if you were to look that up in the table, that codes for the amino acid tyrosine. So this protein at this spot should have the amino acid tyrosine. So here we have a point mutation, 3 different kinds of point mutations. And in this one, instead of being ATG, this last base has been changed to A. So it's not ATG as it's supposed to be, it's ATA. And when you make the messenger RNA from that, it's UAU. But when you look UAU up in the genetic code table, it also codes for tyrosine. So even though your gene has mutated and you've changed one of the bases, you're still going to put the same amino acid in your protein. So that's called a silent mutation because it's not really going to have any effect on that protein's function. Now, instead of changing to ATG, we change to ATC. So the G has now been changed to a C through a mutation. When you transcribe that, we get UAG, and UAG is one of the stop codons. So instead of putting the amino acid tyrosine in when you're making the protein, you're going to stop making it when you hit this mutation. And so you're not going to get your whole protein made. That's called a nonsense, because the protein that's made doesn't make sense. Then the final type is from ATG. This time we're changing the first base to a G, so it's GTG. When you make RNA from that, it's CAC. When you look that up, you get histidine instead of tyrosine. And so when your body's making this protein in the cell, it's going to put histidine at that spot instead of tyrosine. That may or may not have a detrimental effect. It depends how much the way the protein works is altered by the amino acid being different. Remember, when we talked about proteins, we said they fold in specific ways, and that's important for how enzymes work, how structures work. And so if the amino acids are different, it may fold differently and not function properly. So this is called a missense. I always thought these words, definitions were kind of weird, but anyway. So those are 3 types of point mutations. Just to give you an example, we talked a little bit earlier when we did genetics about sickle cell anemia. And sickle cell anemia is a defect in the gene for hemoglobin, which is the protein in your red blood cells that transports oxygen. So normal hemoglobin, on the 6th amino acid, there should be the amino acid glutamate. With sickle cell anemia, that codon in the DNA, it should be GAG, but there's a mutation and the A has been changed to a T. And so instead of being GAG, it's GTG. And GAG codes for the amino acid glutamate, but GTG codes for the amino acid valine. And when you change to valine instead of glutamate, the hemoglobin no longer folds properly and it forms abnormal shapes. That's how you get these sickle red blood cells like you see here instead of the normal shaped. And they stick together, don't fit through capillaries properly. So all of the abnormalities associated with sickle cell anemia, which is a very serious disorder, is due to a point mutation where you have the amino acid valine instead of the amino acid glutamate in the hemoglobin change. These are 2 good videos which I would normally show you in class about sickle cell anemia. If they would work on your computer, you could look at them. They're pretty interesting. So what causes mutations? Many mutations just happen normally. When you think about it, remember how we said in the S phase of the cell cycle, you copy all of your DNA and you have billions of base pairs. And we're actually really good at, if we make a mistake, correcting it. But sometimes a few mistakes get through and that could result in a mutation. Again, sometimes it could be a mutation that gives you an advantage, but it could also be a mutation that is harmful. In addition, there are other agents that can harm your DNA and cause changes in its structure. Some chemicals can do that. Radiation, like exposure to radioactivity, can damage your DNA. Ultraviolet light, which is a big factor here in Arizona with all of our sun exposure. Ultraviolet light can damage DNA in your skin cells, and that can potentially lead to cancer, skin cancer. Viruses, they get inserted into your DNA, so that can disrupt. So say you have a gene and now you have this virus gets hooked into the middle of it, so it's not in the proper sequence of bases anymore, and so that can result in abnormality. And there are some mutations that you can inherit. If you get exposed to ultraviolet light and you get a mutation in one of your skin cells that can lead to skin cancer, that is not inheritable. That would happen to you, but you would not pass that mutation on to your children. The mutation has to occur in sperm and egg cell, have to be present in the sperm and egg cells to be able to be passed on. But there's some that are. You probably have heard there's a breast cancer gene that is inherited. You can be tested for that to see if you have it. If there's breast cancer in your family, they can do a test to see if you have the genetic gene. It doesn't, it's different than like the gene for Tay-Sachs or PKU. If you have it, you have it. But with cancer, they're multifactorial. So if you have the gene for breast cancer, it means you have a greatly increased risk, but it's not 100% that you're going to get breast cancer. This is also a good video, which I would show you in class, about a couple who actually, another inherited type of cancer is one type of colon cancer, and that is present in the dad's family. And so they have do in vitro fertilization, and when they get fertilized embryos, they check by removing one cell and check for the mutations to make sure they're not passing that gene for colon cancer on to their offspring. So it's an interesting video if you would want to take a minute and watch it. This is just showing the ultraviolet light with skin exposure. It causes bases to thymine. If there's two T's next to each other, they connect to each other instead of across to their corresponding to bases. So you don't get normal base pairing, and then that presents a problem when you're making copies of your DNA. That's just an example. That's the end of the protein synthesis. You will now be ready to take protein synthesis quiz 2. And our next topic is histology, which will start on the next PowerPoint. Bye-bye.